SwayamPrabha. Digital India. Educated India.
Welcome back. In lecture 27, we will talk about water quality and testing part 2. So, in this lecture, we will cover the physical analysis for water, chemical analysis and then biological tests for water. So, in physical analysis, the first thing that we check is temperature, and as you know, temperature is measured through a thermometer. And usually we require temperature of water because temperature is very important to understand the density of water, the viscosity of water, vapor pressure, saturation value for solids and gases, and rates of chemical, biochemical and biological activity in water, of course. So, this is where temperature plays a very important role. So, it is better to, that is why we also record that whenever we collect a sample, we record the temperature of water as well. So, temperature is measured, and for public supply, the desirable temperature is around 4.4 degrees centigrade to 10 degrees centigrade, and above 26 degrees centigrade the water is undesirable. That means you will not feel comfortable to drink that water, and above 35 degrees centigrade it is unfit for public supply. So, temperature plays a very important role, and it has to be measured using a thermometer. And then after temperature, the next physical test that we conduct is to check the color of water. And as we have discussed earlier, the color of water is because of presence of organic matter, and particularly in colloidal condition, but it could be also due to mineral and matter as well as dissolved organic and inorganic, other dissolved organic and inorganic impurities. And usually when we determine color, the thing is the color, what color it is actually determines also what kind of impurity is also present. So, that also we can utilize that property as well to determine what kind of impurity, but for that we also have to mix certain other chemicals as well into the water. So, color is determined first by removing total suspended matter using centrifugal force and then comparing with standard color solution or color disk. So, that means there are standard color solutions or color disk, and we first need to remove the suspended matter from the water, and then whatever is the remaining material, remaining water, for the color of that is actually measured. And standard unit of color is that which produces what is produced by 1 milligram of platinum cobalt dissolved in 1 liter of distilled water. So, that is 1 ppm. So, 1 milligram of platinum cobalt dissolved in 1 liter of distilled water actually gives a color which is called the standard unit of color. So, usually in domestic use we allow 20 ppm is the permissible limit for this color. So, that means it is sort of a relative measure, you can understand that. So, we have this color disk that we can get, and concentration of chlorine, pH, iron, silica, nitrogen, all these things can be determined by adding reagent into the water and then turning the wheel until the sample matches the reference color, right. So, we can mix some reagents with water and then can test it using this color disk, or directly we can test the existing water and its color and see what it matches to determine what kind of impurity it is. In addition, we also have got these photometers which we can, these are portable ones which you can carry along with you, and this color of and this can actually measure color of water from 0 to 500 PCU, which is platinum cobalt units. So, this kind of water testing kits could be these meters could be also taken in the field where we can measure color directly. Then comes turbidity. And turbidity, as we have discussed, is due to the presence of suspended and colloidal matter, and water appears muddy, cloudy or turbid, and the unit of turbidity is milligram per liter or parts per million, both of them, and standard unit is produced by 1 milligram of finely divided silica in 1 liter of distilled water. So, that is the standard unit that we use, and excess of 5 units is actually easily detectable. That means, when the turbidity limit is more than 5 units, we can detect it, and permitted limit is around 1 to 10 units of turbidity, and we use a turbidity rod or a turbidity meter to determine the turbidity of a particular sample. Now, this turbidity rod is basically an aluminum rod, and it has got a platinum needle fitted in front of it, and when we dip this particular rod into the water, and at certain level you will see that the needle is not visible to the eye, and that is this particular aluminum rod is graduated, and based on that reading of the graduated rod on the surface of the water, wherever there is at the water level, so that is the value of turbidity of that particular sample. So, this is how we can determine turbidity using a turbidity rod. But in addition, we can also use a turbidity meter, and then again you can carry this in the field again, and this is basically a turbidity meter measures the scattering caused in the water sample due to passage of light rays. Because of the presence of these impurities or suspended particles inside that particular water and colloidal particles inside water, what happens, the light rays get scattered, and we need to measure that. If that could be measured, based on that we can actually determine what kind of turbidity is there in the water. So, there are different kinds of turbidity meters, such as Jackson turbidity meter, Baylis turbidity meter, Hack laboratory turbidity meters, and nephelometers, of course. So, usually what it does is, in case of this Hack laboratory turbidity meter, intensity of passing light is measured using a photometer, and similarly, as you can see in this particular picture, we can put the water in this particular container, and then the system can put on a LED light, usually a blue LED sometimes, and then intensity of the reflected light is used to measure degree of turbidity in the turbidity meter. So, we put on a light and we measure the intensity of the light on the other side, and we can understand based on the values that we read the turbidity of that particular sample. So, then comes test and order. So, these are other properties or physical properties of water, and usually this is because of dissolved gases such as H2S, CH4, hydrogen sulfide, methane, carbon dioxide, oxygen, and then mineral substances like sodium chloride, like salt, iron compounds, carbonates, sulfates, or due to some presence of some other organic matter. So, these are the ones which gives both test as well as order, and usually we determine their usually order is measured by order intensity, and there is something called a threshold order number. That means, we have a sample, and then the sample is diluted with order free normal water, and the number of times the sample gets diluted actually gives us the threshold order number. So, if a sample requires 3 times its volume to to mix fresh water to be mixed with it to become undetectable, that means there is no order coming out of it, then that 3 would be the threshold number of that particular sample. So, for public supplies, 1 to 3 is the threshold order number for the water that needs to be supplied. So, similar to test and order, we can also measure the specific conductivity of water, and this actually gives us an idea about the kind of dissolved salts that are present in the water, and this is done with a portable Dionic water tester. What it does is it measures the sort of the kind of the current inside the water or the charge, and like the unit of measurement is MHO. This is opposite of ohm, and this is measured 1 MHO is 1 ampere by 1 volt, and MHO per centimeter at 25 degrees centigrade is measured, and that is multiplied with a coefficient which is 0.65, and that gives us the quantity of dissolved salts in the water. So, why we need to measure all this quantity of salt? Because based on the quantity that we measure. Based on that, we have to determine in our treatment units what kind of maybe chemicals that we have to mix with our for treatment of that particular water, or what kind of treatment sequence we have to put in so that we can get rid of that kind of salt, that amount of salt, right. So, that is why we need to measure this, and specific conductivity of water, as you can understand, like over here you can see this portable conductivity meter, where we can test both for salinity and TDS could be also measured in this one. And here there are 4 platinum rings are used in this particular unit. So, this is basically the meter, the what to say, the interpreter of the results, but this is where the actual testing takes place. This is the unit which is actually attached with this particular unit. So, this is the sensor, you can say, in this particular case. So, this has got around 4 platinum rings. Top and bottom rings outside apply alternating voltage and induces a current. Center ring inside measures the potential drop in the current, which gives an electrical conductivity reading. So, that is how we measure conductivity of a sample, and this presence of salt is basically could be computed from that, and this actually represents the kind of salinity and also TDS. This could be determined from this one. So, then we look into the once physical analysis is done, then we also need to conduct some chemical analysis. The one that we conduct, the most important one, is to determine the pH value of water, because it influences a lot of processes. And pH value of water indicates, as you know, that it is the logarithm of reciprocal of hydrogen ion concentration in water, and this indicates that if a water is actually acidic or little bit basic or alkaline, right. So, as you know that water is weakly ionized with H2O is equal to H plus plus OH minus, and we know that from the law of mass action of physical chemistry that concentration of H plus ions into concentration of of OH ions divided by concentration of undissociated HOH molecule is basically a constant, which is 10 to the power minus 14. So, usually we assume that pH value of neutral water is basically where H plus and OH minus ions are of same order, that is 10 to the power minus 7, and pH value is basically the log of that of the reciprocal of hydrogen ion concentration, which comes to around 7, right. So, pH value of neutral water is 7. So, more than that, that is 0 to 7 is the acidic range, and 7 to 14 is the alkaline range. So, if it is more than 7 to 14, that means more amount of OH ions are present, that is why it is acidic and basic or alkaline, or if it is less than and if it is less OH ions are present, then it is or more H plus ions are present, then it is acidic, or the pH value would be 0 to 7. So, usually when we add acid to water, that means it becomes acidic. So, we have got more H plus ions, because they will increase, acid will increase H plus ions, whereas in case of NaOH, alkali will be increased because of OH sodium hydroxide. So, OH ions are there. So, this will actually increase, and this will, the sample will become alkaline. So, how do we determine pH values? So, there are different methods. So, there is the colorimetric method where indicators are added. As we were discussing earlier, we can add some reagents to the water, and color so formed is compared with standard colors which are there, and we can determine if what kind of what is the pH value of that water. And electrometric method is special electrodes are dipped into the water, and the meter can determine what is the pH value, and lower pH value causes tuberculation and corrosion, whereas high values produce incrustation, sediment deposit, difficulty in chlorination, etc. So, that is why pH, we need to be careful about pH value. That means, if the pH value is lower, which is acidic, that it will create tuberculation and corrosion of the pipelines, right. Tuberculation is one form of corrosion, of course, and then whereas high values of that creates incrustation, that means sediments, some deposits happen, right, and it also creates difficulty in chlorination, and this kind of sensor units could be utilized. Whereas, as you can see, this actually gets fitted into that handheld device. So that handheld device could be common. We can change this kind of sensors, and using this sensor we can use pH. Similarly, using another sensor we can determine conductivity and so on. So next, in chemical analysis, we determine the total solids or the total amount of suspended solids, and we determine that through filtering and then drying and then weighing it. And once the suspended solids are determined like that, like we have conducted in schools, we have also conducted experiments where we take a filter paper, we put in the solution, whatever is caught on the filter paper is actually the suspended solids, right? And we can dry that particular suspended solids or that filter paper, we can get those suspended solids out of the paper, and we can weigh that to get a total amount, volume or total weight of that particular suspended solids. Whatever is left which is not being caught by the filter paper, that we can get by evaporating the remaining water and weighing the residue. So this actually contains both dissolved and colloidal solids, and this residue could be further heated on a furnace where organic solids will decompose and only the inorganic solids will be left, right. So these are the two ways we can determine what is the total amount of solids in a particular sample. Then is hardness. Hardness in water, we have again learned about this in 10+2 levels, and you can know that hardness is that characteristics of water which prevents formation of leather or foam when mixed with soap, and this is because of the presence of calcium and magnesium salts, and these forms come by reacting with soap, and that is why the soaps are not that effective. So hardness of water also causes greater soap consumption, but at the same time it also causes scaling of boilers and causes corrosion as well. And there are two kinds of hardness. One is temporary hardness and other is permanent hardness. Temporary hardness is because of carbonates and bicarbonates, calcium and magnesium carbonates and bicarbonates, and we can easily get rid of that by just boiling or adding lime to the water. But in case of permanent hardness, it is difficult to remove, and this is mainly because of sulfates, nitrates, chlorides of calcium and magnesium, and this requires specialized treatment or water softening treatment. And usually hardness is measured in milligram per liter or ppm of calcium carbonate in water, and we actually determine this by the Varsante method, where EDTA salt solution is actually used, and when this hardness is there, then red actually this salt solution converts from red to blue color, and then we can detect the hardness value, and hardness levels up to 75 ppm are considered as soft, and when it is 200 ppm it is considered hard. So as you can see that we measure the calcium carbonate in water, so until 75 ppm it is considered soft, but when it is more than 75 ppm up to 200 ppm and above, then we will consider it as hard. Then other chemical analysis involves chlorides, and sodium chloride is the main substance in chloride water. Sodium chloride gives that results in presence of chlorides, and this could be because of mixing of saline water or because of mixing of sewage in water. So if we find chlorides in water, then we understand that either saline water has mixed or sewage has mixed, and above 250 ppm is not permissible at all. So we should be careful about that. Then chlorine, usually as we have already discussed, chlorine is used as a disinfectant, and there is residual chlorine in water, in distributed water. That means after treatment, the water that gets distributed, we usually find residual chlorine in that from 0.5 ppm to 0.2 ppm that is left in water to make the water bacteria free, pathogen free, and we can determine this through a starch iodide test, and where the quantity of chlorine in milligrams per liter in water sample is equal to 0.355 into the number of milliliter of thiosulfate required to remove blue color through titration. So basically in the starch iodide test, when this potassium iodide and starch solution is added, the water turns into blue, and when then we keep on mixing the thiosulfate, and then gradually we mix that through titration, and when the water the blue color is removed, and we measure that amount of milliliter of thiosulfate, we multiply that by 0.355 and we can get the quantity of chlorine in water. Too much of chlorine is also not, it gives a bad taste, and it sometimes results in some other issues. So, that is why too much amount of chlorine is also not suggested. Then iron and manganese usually found in groundwater, and above 0.3 ppm creates, particularly for iron, creates brownish red color, and this creates stains in cloth. It also corrodes pipe, and some test and order issues are also the results because of this, and this could be measured using the calorimetric method by comparing with standard color solutions, same as earlier. And finally, we have got lead and arsenic, and lead from pipes and arsenic from groundwater. Sometimes in very rare cases are available, and like in Bengal, we find in some certain districts, we find arsenic in the groundwater that is pumped. So, we have to be careful, and those kind of tests has to be conducted. Then nitrogen content in water. Presence of nitrogen in water indicates presence of organic matter in water, and this also indicates presence of bacteria. And there are different kinds of nitrogen which are present in water. For example, there could be free ammonia, which actually indicates the first stage of decomposition, and the limit is around less than 0.15 milligram per liter. And usually when we boil this, then this releases ammonia gas, and this could be measured using the distillation process, the quantity of that, and from that we can determine the amount of free ammonia in water. Then nitrates, this actually indicates partly decomposed oxidized organic matter. It should not be present, so limit is nil, and it is again determined through color matching method by adding sulfonic acid and naphthamine. Then nitrates, this indicates fully decomposed or oxidized organic matter lesser than 45 milligrams per liter, and again determined through color matching method by adding phenol disulfonic acid and potassium hydroxide. And then organic nitrogen, this is the quantity of nitrogen before decomposition, and limit is 0.3 milligram per liter, and this could be determined by adding potassium permanganate to boiled water and then boiling that. So, and this boiling will release ammonia gas, which could be measured, and from there we can measure the quantity of this nitrogen. Now, the thing is, even though there are different kinds of nitrogen, nitrogen is available in different forms, nitrates, nitrites, ammonia, organic nitrogen, and there are different tests for all this, and usually some of this test has to be done at the laboratory level, but sometimes we can carry these small kits which are available in the market, and they have these different solutions that are mentioned over here, and you can take them along with you when you are going for sample connection, and you can do this kind of test over there in the field itself, and that will actually save time, because sometimes we don't have 72 hours to come back to the laboratory and do this kind of testing, and some of this you may not do all the test. This could be some of these tests are indicative, like if you get already this nitrate level is nil, so usually we will probably not find the other values as well, other kind of nitrogen as well. So, this kind of test could be, it is better to conduct in the field directly, and for that we need to carry this kind of testing kits along with us. So, other chemical analysis involves dissolved gases, where nitrogen, methane, hydrogen sulfide, carbon dioxide, oxygen, these are the dissolved gases we usually find, and CO2 indicates biological activity, converts carbonates to bicarbonates, and oxygen usually keeps the water fresh and sparkling, and oxygen is usually absorbed from atmosphere and is consumed by organic matter for oxidation. If less than saturation level, then this indicates presence of organic matter. So, this is very, very important in later lectures. you have heard us talking about BOD, and we will talk about the biological oxygen demand and all. So whenever there is impurities in water, biological impurities, there is the water gradually gets treated, and these BOD levels actually help us to determine if the water is completely treated or not. So if less than saturation level is present, then this indicates that there is organic matter. That means some amount of biological oxygen demand has to be yet to be satisfied. So we measured the dissolved oxygen in water, and that is very, very important, which gives us an understanding about if the water is safe for drinking or if it is with contaminated with certain kind of impurities which needs to be first treated. So dissolved oxygen levels can be measured using Winkler titration method or electrochemical analysis method. And in this particular method, we use electrodes to detect the amount of oxygen when it passes through a highly permeable diaphragm. It permits oxygen, and we can measure that. And we can also measure using photochemical analysis method or fluorescence method. Fluorescence basically is a material which is getting excited by light, and we usually use a blue light emitting diode, and when oxygen passes through a DO permeable layer, that is dissolved oxygen permeable layer, then we can measure that using a diometer. So you can see this is a diometer where optical sensors are used for determining the luminescence, and usually when we dip this in water, then again we can determine what kind of DO value is there in water. So biochemical oxygen demand is, as I was discussing, it's the amount of oxygen at a specified temperature under aerobic conditions required by bacteria and other microorganisms while decomposing organic matter. So organic matter is acted upon by bacteria, and they are gradually decomposed. So once the entire decomposition happens, then water is safe. But till that happens, we can determine that by determining the biological oxygen demand. So BOD is tested for both raw and treated water, and usually for treated water BOD would be 0 because all the biological activity is taken care of. But what we do is, whenever we are taking a sample of water, we measure the dissolved oxygen of that particular water, and we also then measure it 5 days after at 20 degrees centigrade. We store the water at 20 degrees centigrade, and we measure the DO levels at at the end of 5 days. And the value of BOD is actually the standard value, or the BOD of water during the first 5 days at 20 degrees centigrade is taken as the standard demand or biological oxygen demand. So as I was telling you that we take 2 samples at each site. One is first immediately tested for the dissolved oxygen, and the second sample is kept in the dark at 20 degrees centigrade, and we test after 5 days we will test it for the remaining oxygen or the DO values. The difference in the oxygen level in these 2 tests at least gives us the amount of BOD in milligrams per liter. So if the first sample result was 10 and the next result was something else, we take the difference between these 2, and that actually gives us the biological oxygen demand for this particular sample. So finally, we conduct other biological tests involved determination of bacteria, viruses and protozoa, which usually infect human beings. And for that we do total count of bacteria. This only could be done at the lab level. This cannot be done in any field test or by using any meters, but this has to be done at the laboratory level. And we count the total number of bacteria in a milliliter of water under microscope. And we also conduct E. coli test, where we detect the presence of coliform group of bacteria, where both total coliform and fecal coliform bacteria is measured. And these are, as we have discussed earlier, coliforms are non-pathogenic bacteria. These are rod shaped, but the presence and absence of this bacteria actually indicates there has been fecal pollution, and hence because of fecal pollution there may be other pathogens, right. So E. coli is the predominant member of the fecal coliform group, and presence of E. coli actually indicates that there is fecal pollution. But instead of E. coli, we can also measure the total coliform or fecal coliform, and that I also will do, and these are again could be only measured at the laboratory level. So, to conclude, water needs to be tested to determine its characteristics and to determine the quantity of impurities in it. While some of these tests needs to be conducted in a laboratory setting, many can be conducted at the field. Then temperature, pH, dissolved gases, etc. is determined on the spot during sample collection, and portable handheld meters are now available for many of the recommended tests. These are the references. Thank you.